Before starting the experiment, we prepare all the required apparatus. These include sample cell for nitrate and nitrite from both point sources A and B, which is A is from inlet and B is from the outlet, as well as blank sample cells for calibration. We also use measuring cylinders, nitrile-6 and nitrile-3 reagent powder. To prepare the blank sample cells, we measure 15 ml of distilled water using a measuring cylinder and pour them into a separate sample cell. These blank sample cells are essential for zeroing the spectrophotometer to ensure accurate readings. We begin the nitrate test with the water sample from point A, which is the inlet. First, 15 ml of the sample was measured and poured into a sample cell. Then, we add nitrile-6 reagent powder, and the sample cell was shaken carefully for 2 minutes. After the reaction, 10 ml of the treated sample was extracted using a syringe and transferred into a new sample cell. Nitrile-3 reagent was then added. The cap was sealed and the sample was shaken again for 3 minutes. A pink color began to form, indicating the presence of nitrate. For the nitrite test, 10 ml of the same water sample from point A was used. Only nitrile-3 reagent was added, and the sample cell was shaken thoroughly. A similar pink color appeared, indicating the presence of nitrite in the water. We then used DR5000 spectrophotometer to measure the readings of each sample. Before inserting any cell, we wipe it clean to remove any residue or fingerprints that could affect the results. We first inserted the blank sample and zeroed the spectrophotometer. The reading obtained for nitrate was 0.08 mg per liter. The same procedure was repeated for nitrite, and the value we obtained is 0.048 mg per liter. The procedure is repeated for water sample from point B. Our next experiment is turbidity test. We conducted a turbidity test to evaluate the clarity of the water samples collected from CEEC section 7. First, we pour the water sample into a clean beaker. Then, we transfer the sample into a turbidity meter cell, carefully filling it up to the mark line to ensure accurate measurement. We then wipe the turbidity meter cell by using a towel to ensure there is no dirt or fingerprint. Okay, now we turn on the turbidity meter and press the signal average button. After that, we adjusted the range setting until the screen display auto range and signal average, confirming that the device was ready for measurement. We inserted the turbidity meter cell and pressed the button read. After we get the value, we repeat for other sample.
To begin, prepare a blank sample by pouring distilled water into a clean beaker. Using a micropipette, take 2 ml of distilled water and pour it into a low-range vial. After that, close the cap of the vial and gently shake it to mix the contents. As you can see, these were the water samples that my group used. The first sample was from the inlet, and the second sample was from the outlet of the CEEC section 7 Shah Alam. For the sample 1, pour the water sample into the beaker. Using a micropipette, take 2 ml of the water sample and transfer it into the low-range vial. Close the cap of the vial and gently shake it to mix the contents. For the second sample, the same steps were repeated to conduct the second water sample. Pour the water sample into the beaker. Using a micropipette, take 2 ml of the water sample and transfer it into the low-range vial. Close the cap of the vial and gently shake it to mix the contents. The COD reactor preheated will calibrate and prepared. All the prepared vials were placed into the preheated COD reactor at 150 degrees Celsius for 2 hours for digestion. After digestion, the vial were carefully removed and left to cool at room temperature for approximately 45 minutes. Each vial was placed into the device to measure the COD concentration. The appropriate program for COD 430 low range was selected. Wipe the bottom of the vial before inserting it into the spectrophotometer. After that, place the blank sample into the instrument, then tap the 0 button to zero the reading. Wipe the bottom of the vial for water sample 1 before inserting it into the spectrophotometer. After that, place the vial for sample 1 into the instrument to obtain the reading. The same procedure was repeated for the second water sample. For the color test, the 2 water samples were prepared in 2 separate beakers. Each water sample was poured into the sample cell. Using the DR2800 spectrophotometer, calibrate the instrument and select the appropriate program for the color test. Wipe the bottom of the sample cell with a towel to avoid any reading errors. First, place water sample 1 into the instrument, followed by water sample 2.
Hi everyone, today I will presenting the step-by-step procedure for conducting the biochemical oxygen demand or BOD test. We begin by preparing 2 liters of dilution water. Into this, we add 2 ml of each of the following chemical reagent: phosphate buffer solution, calcium chloride solution, ferric chloride solution, and magnesium sulfate solution. Next, the oxidation process begins. This is done by aerating the dilution water for 1 hour to ensure it is fully saturated with oxygen. While waiting, we label our BOD bottles accordingly to match each water sample. Each bottle is then filled with 150 ml of the sample water. The remaining volume is filled with the prepared dilution water until it reaches the neck of the bottle, making sure there is no space left for air. A stopper is carefully attached to each BOD bottle to ensure no air bubbles are trapped inside, as this may affect the accuracy of the test. We then switch on the DO meter and clean the probe with distilled water. This step is important to minimize contamination and ensure accurate reading. The clean DO probe is then inserted into the BOD bottle, and the magnetic stirrer at the probe is turned on to mix the sample gently. We wait for the DO meter to beep or display the asterisk symbol. This indicates a stable reading, and we record the initial dissolved oxygen value, which we refer to as DO0. Finally, the remaining BOD bottles are stored in a refrigerator or incubator at a controlled temperature of 20 degrees Celsius. We wait for 5 days, after which they measure the final dissolved oxygen level, known as DO5. This completes the BOD testing procedure. Thank you. Now, I will demonstrating the testing for the pH level. First, we label our water sample at sample point 1 and sample point 2 to ensure clarity. Next, we clean the probe of the pH meter using distilled water. This is an important step to remove any previous residue and ensure accurate result. After cleaning, we gently insert the probe into our first water sample. The probe must be fully submerged for an accurate reading. Then we adjust the probe so it is steady and not touching the side of the bottom or the container. Once it is in place, we switch on the pH meter and allow the reading to stabilize. When the pH value is steady, we record it. We repeat the same procedure for sample point 2, and that's how we perform a pH test using a digital pH meter.